Hello, my name is Ariana Nava, and today I will be presenting on the mutation HOX13 and Brachydactyly Type D. So what is brachydactyly? This is a condition that affects both the toes and fingers, causing them to be short and stubby. Specifically today, I will be talking about the type D, best known as the short thumb. This is when the distal phalanx, referring to the middle bone of your thumb, is shortened and widened. To the right, we have an image of an x-ray of what that looks like. This condition is autosomal dominant, meaning only one parent needs to carry the gene in order for it to get passed down to the offspring. Both genders are usually affected, but it is most commonly seen in females. It is usually bilateral, meaning that both thumbs are affected, but in rare cases, it can also be unilateral, meaning that only one thumb is affected. This condition isn't life-threatening. Most people with it live a healthy life. Surgery may be done if desired, depending on the severity of your case. So what mutation is responsible for this? HOX13 is a homeobox protein coding gene that may be involved in Brachydactyly Type D. HOX13 is a missense mutation in this condition. It plays a role in embryonic development. HOX13 is responsible for sending a transcription factor involving important functions in limb patterning and growth, which is why mutations in this gene give rise to limb malformations. To the right, we have a reference picture of what the condition looks like. This is one of those rare cases where the individual is unilateral. Their left thumb is the affected one here. The first article that I will be talking about is called Missense Mutations in the Homeodomain of HOX13 are associated with Brachydactyly types D and E. The methods used in this article were linking HOX13 to Brachydactyly in humans. HOX13 was amplified by PCR for 128 patients with limb abnormalities present at birth. The PCR test was testing for the organism to see if the HOX13 gene mutation was present in affected families. To the right, we have a family pedigree of family C, which is involved in the research. We see that this family carries a gene and it is autosomal dominant because we see it present in every generation. We also see that females are most likely to be affected. DNA sequencing and mutation confirmation was used to identify two mutations responsible, these two. So the results from sequencing two HOX13 mutations to Brachydactyly type D are that family C confirms that HOX13 is responsible for brachydactyly. This is because when they sequence the two, we see that the amplification for the normal individual is normal. And then when we see the amplification for the affected individual, we follow the arrow, we see that there's no amplification in the beginning, indicating that there is a loss mutation here. This is a site that indicates that there is no mutation. The 940A to C mutation was confirmed by blot hybridization using the allele-specific oligonucleotide. This is because this is referring to this right here. The affected individual doesn't have it, meaning that it was blocked. The second method used in the first article is DNA binding aside. Authors speculated that HOX13 missense mutations would affect DNA binding using electrophoric mobility shift asides to test this. They tested the binding of wild type and mutant proteins. The ARG320 ALA served as the negative control, important for residue for contacting with the phosphate backbone of DNA. And then the positive control was the ILE314 LEU served as the variant. The results from the ILE317 LEU variant increased binding to DNA. No DNA binding was found in the negative control protein, which is this one, but binding did occur in the second and the positive control right here. We see it present in these probands right here, indicating that the positive control group has greater binding. We also see that affinity for the positive control group increases right here with these. And then in the first negative control, it decreases. The second article that I will be talking about is called Mutant HOX13 Induces Extra Digits in a Mouse Model of Sympolydactyly Directly and by Decreasing Retinoic Acid Synthesis. Researchers expected the mutation to have an effect on limb development in mice. The methods used here were linking HOX13 mutations in mice to compare to humans. The SPDH mice served as a model for humans. The RA measurement was used. So right here to the right of the image, we see that the wild type right here has normal fingers. They have normal growth. We see that the fingers are growing correctly. And then the mice that were infected, we see that their fingers are starting to grow deformed. So the results are that HOX13 is required for limb formation. The control group had normal limb formation as compared to the mice. The limb appears together. In the experimental group right here, we see the bones in the thumb starting to get shortened already. And then in the wild type, we see normal growth. The wild type shows a normal growing rate, while in the SPDH, we see the HOX13 is affecting the limb development. The HOX13 allele resulted in a distinct phenotype, right? Just like referring to the thumb. Conclusions are that HOX13 is required for limb formation and growth. Limb formation is required for the correct shape of the thumb. The RAM mutation of 940A-C causes loss of function in HOX13. Loss of HOX13 protein expresses Brachydactyly Type D in a heterozygous family carrying the gene. The loss of function in 940A-C variants may cause Brachydactyly Type D through loss of limb formation. The main conclusion is that linking the three results together provides a possible mechanism for how the mutation in the HOX13 specifically results in altered thumb structure. Future directions include determining the location of the 940A-C mutation in a larger study or different population in affected individuals with the condition, to determine if the variant encoding ILE314 LEU is required for limb formation in other organisms, determining the pathway of the mutation, determining if other possibilities might lead to Brachydactyly Type D, such as blood flow problems to the hand and feet, especially in developing babies. This is my reference page. Thank you.